For decades, scientists have relied on a genetic map to navigate one of the most important organisms in biology. But here's the kicker. What if that map had huge, uncharted territories? Today, we're unfolding the story of how that map was finally, almost, completely filled in.
We've all seen them, right? Buzzing around the fruit bowl. But in the lab, these tiny insects are absolute superstars. I mean, since the early 1900s, the fruit fly has been a go-to tool for geneticists. And why is that? Well, it's because so many of its genes have direct counterparts in us, in humans. That makes it this incredible model for studying everything from how we develop to how diseases work.
And that really gets to the heart of it, doesn't it? For all the years we've been studying them, our guide to the fruit fly's DNA, its genome, was kind of like one of those ancient maps of the world, with vast areas just marked, "Here be dragons." We knew there was more out there to discover, but we just couldn't see it, yet. Okay, so our story of mapping this genome really kicks off with this monumental achievement right at the turn of the millennium. So back in the year 2000, there was this massive collaboration that gave us the very first draft of the fruit fly genome. It was a landmark moment for sure, building on a whole century of research. But as you can see from this timeline, that was really just the beginning of a long, long journey of refinement. So you might be wondering what exactly makes the fruit fly such a perfect model organism. Well, it's a few things. It has a really short life cycle, it's super easy to raise in the lab, and its genetics are relatively simple to mess with. This lets scientists run experiments and then apply what they find directly to more complex systems, like, our own bodies. And the real reason they're so perfect for this, it all comes down to the numbers. The fruit fly has about 13,600 genes. Humans, we have somewhere between 20 and 25,000. It's not a crazy difference. And the incredible thing is that a huge chunk of those genes are shared between us. This genetic similarity is exactly what makes the fruit fly such a powerful stand-in for understanding ourselves. Okay, so that first genome sequence in 2000, total game changer. But it was still just a draft. So the next chapter in our story is all about turning that rough sketch into a much more detailed, much more accurate chart. And that map analogy, it really is perfect. The early version had what scientists call gaps. Basically, these were blind spots, stretches of DNA that the technology back then just couldn't read. And these gaps often showed up in the most complex, most repetitive parts of the genome, hiding who knows what kind of important information. Fast forward to 2014, and wow, the technology had gotten so much better. This allowed for a major update. Just look at the massive leap they made with release 6 of the genome. The total size grew, the gaps got a lot smaller, and check this out, the Y chromosome is over 10 times more complete. That's huge. And it was possible because they could now focus on sequencing this really tough stuff called heterochromatin, which is just a fancy term for dense, tightly packed DNA that's a real pain to read. But here's the thing. Even with all that amazing progress, the job still wasn't done. The Release 6 map, which was the gold standard for years, still had about 250 of those unresolved gaps. 250 blank spots still on our map, just waiting to be filled in. And that, my friends, brings us to today's big breakthrough. It all came from a new era of technology, especially something called long-read sequencing. So instead of trying to piece the genome together from millions of tiny little fragments, scientists could now read much, much longer stretches of DNA at once. It's like going from a 10,000-piece puzzle to a 100-piece one. This allowed them to create a near-perfect telomere-to-telomere map, which just means they mapped each chromosome from the very tip of one end to the very tip of the other. And just look at the difference here. It's staggering. The new version, called DMTT2, is way bigger. But the most important number here is the gaps. It plummets from over 250 all the way down to just 18. Even the N50 got better, which is basically a score for how complete and unbroken the genetic map is. This is as close to a perfect picture as we have ever had. Let that number sink in for a second. Over 93% of the remaining blank spots on the map were filled in one go. That is a truly massive leap forward for genetics. Okay, so the big question is, what was actually hiding in those gaps all this time? Turns out it was more than just junk DNA. We found entirely new biology. And probably the most surprising thing was a type of mobile genetic element, a jumping gene, called a sign element. For decades, the consensus was that these things simply did not exist in fruit flies, and yet there they were, hiding in the gaps the whole time.
Now, this chart really puts that discovery into perspective. Signs make up such a teeny tiny fraction of the genome, just 0.02%. It's barely even visible here. But their presence, no matter how small, completely rewrites a small part of the genetics textbook. It's a perfect example of how filling in the map can lead to brand new discoveries.
All right, so we have this nearly perfect map. Why does it matter so much? What's the big deal? Well, it's not just about cleaning up the data. It's about unlocking a whole new wave of potential discoveries. Well, the impact is immediate and it's huge. Researchers found 92 genes that were completely unknown before. They could finally map the really complex ends and centers of chromosomes, the telomeres and centromeres. They could compare different strains of flies with incredible precision. And, crucially, they could start linking these newly discovered genes to real biological functions, like the sense of smell. And you don't have to take my word for it. The researchers who actually did this work said it best themselves. This isn't an endpoint. It's a powerful new resource. It gives scientists everywhere this stable, high-quality foundation for countless future experiments, letting them explore the fruit fly's biology in a way they never could before. So here we are. The map of the fruit fly genome is more complete and more detailed than ever before. But the age of exploration? It's not over. In fact, you could say that with this new guide in hand, it's really just beginning. The real question now is, what new secrets are we going to unlock next?